Welcome, everyone. Great to see you here. And we are going to start right now with the fourth virtual session of the Xylella fastidiosa workshop, From Diagnosis to Management. This workshop was organized in partnership between the Escola Superior de Agricultura Luis de Queiroz from the University of Sao Paulo through the Laboratory of Insect Vectors of Plant Pathogens, as well as with the Citriculture Center, Instituto Agronómico de Campinas, through the Laboratory of Biotechnology. This workshop has been supported by the Red Iberoamericana para la Vigilancia de Xylella fastidiosa, IBER-XIFAS, which is funded by CITED, which is a Spanish international funding agency. Department of Entomology and Acarology of the University of Sao Paulo, and the graduate program in entomology of the University of Sao Paulo. The workshop is designed for researchers, professionals, growers, and students. And its goals are to provide an updated overview on the plant pathogenic bacterium Xylella fastidiosa, its importance, classification, host-plant interactions, transmission by their vectors, detection, and monitoring, as well as on management and certification strategies. Another goal is to provide training on research techniques related to the pathogen and its vectors. The workshop includes 5 virtual sessions. Today we are going to present the 4th virtual session, and then there will be practical sessions during the days 5 to 7 of September. So right now I would like to introduce the first speaker for today. We have 3 speakers. Every speaker will have 30 minutes. And I am pleased to introduce Rodrigo Krugner, who will present his talk. Hi, Rodrigo.
Good morning, Dr. Fernandez. How are you? Thank you for the introduction. I'm ready to go.
Nice to see you. Rodrigo Krugner, he's Brazilian, by the way. He's working in California. He has a BS in agronomy at the University of Sao Paulo in Brazil, a Master of Science in Plant Science in California State University, Fresno, and also a PhD in entomology of the University of California at Riverside. He began his career in the U.S. Department of Agriculture, in the USDA, in 2007 as a research entomologist in the Crop Diseases, Pests, and Genetics Research Unit at the San Joaquin Valley Agricultural Sciences Center in Parlier, California. He has since that time worked on a variety of projects involving the bacterial pathogen Xylella fastidiosa and associated insect vectors. Related to these projects, Dr. Krugner has conducted research on biology, behavioral, and population ecology of insect vectors and their natural enemies, with major focus on plant-insect pathogen interactions. In addition, Dr. Krugner serves as a research leader for the research unit, where his research is conducted to develop methods to mitigate the impacts of pests and diseases of grapevines and citrus, and to develop new varieties of table grapes and stone fruits. So after his talk, the questions will be at the end of the 3 talks we have today. So I now leave Rodrigo to start his presentation. Thank you, Rodrigo.
Thank you. Can you see my first slide? Can anyone see my slide? Can anyone hear me? Yes, okay, thank you. I saw that on the chat. Well, thank you very much for the invitation to present here today. So today I'm going to focus on the Californian case of Xylella fastidiosa, and I'm going to focus primarily on Pierce disease of grapevines. We do have several diseases caused by Xylella here in California, but today, because of the time, I'm going to focus only on Pierce disease. So just a brief overview of California agriculture. I don't know if you can see the picture on the bottom left there. So California has about 400 different agricultural commodities, about 70,000 farms with an average size of 140 acres. So we're talking about a mix of almond trees and grapevines and berries and strawberries and 400 different commodities. Again, very small farms. Grapevines and almonds are the top two agricultural commodities, well, except for milk and cream. We do have a lot of dairy cows here in California, but almonds and grapes account for most of the cash receipts in terms of a return in the income. Again, with the United States Department of Agriculture, Agricultural Research Service, there are a number of agencies inside the USDA. The agency that I work for is the Ag Research Service. And I don't know if you can see on the map in the United States, those white circles in there. So each state has a research center and they address problems from each location there. So this is a picture of our center here. So it's about 50 hectares. We have about 100 people working here. We have a variety of disciplines: chemistry, entomology, plant pathology, plant breeders, horticulturists, and so on. So this is the outline of my presentation for today. So I'll just give a brief introduction of Pierce disease of grapevines. Then I'll talk about the invasion of glass-winged sharpshooter in California, which is an important vector of Xylella here. Then the control guidelines for Pierce disease here in California. And then I'll go through what's called the CDFA, Pierce Disease Control Program. CDFA is the California Department of Food and Agriculture. That's a state agency that coordinates the program to control Pierce disease. And then I'll finish with some general conclusions. So California vine disease, which is now known as Pierce disease, was first characterized by Newton Pierce. Newton Pierce was a special agent with the United States Department of Agriculture. He was sent to California in the late 1980s to look at a new disorder in the grapevines. So he published this, it's a pretty thick book describing the disease for the first time. And we fast forward to today. Pierce disease costs California about $104 million a year. That's what it costs to the industry to control the disease. There's some research money that goes into there, regulatory issues, and I'll go through some of those today. So it took some time, but they eventually figured out that Pierce disease was being caused by Xylella fastidiosa. So there is a picture of a xylem vessel, the one in the center that has those little rod-like things. Those are colonies of Xylella fastidiosa. So they'll clog the xylem vessels. The ones next to it, they're empty, although Xylella can move sideways on the plant. So Pierce disease diagnosis is based on observation of symptoms and diagnostic testing. And this is very important. Here's some pictures of symptoms of Pierce disease. So on the top left corner, what we call like green islands, sometimes irregular patchy bark maturity. So you have portions of the cane that are brown and some remain green. Next to it, on the right side, petioles remain attached to the canes. Those are usually we call matchsticks. So the leaf blade will fall off. Only the petioles will remain on the vine. And then if we keep looking to your right, symptoms vary depending on the variety. So if you have a red grape variety, those are the symptoms of a red grape in general. And then if you're looking at a white grape variety, those are the symptoms for a white grape variety. Symptoms can be confusing. So usually what it's done, and I was actually looking some emails right now for almond leaf scorch disease. There are a number of things out there that can cause symptoms. They're similar to Pierce disease. So it's important to do some testing in the lab. So culturing is usually done when we're doing research only. There's some private labs that can do ELISA tests and then also PCR tests. So it's very important to confirm presence of Xylella fastidiosa based on those. Again, reason is there are a number of things out there that can cause disease. So on this picture here, there are 3 different types of symptoms. One is Pierce disease, the other one is meals, and the other one is a sulfur burn. So it can be very confusing. So it's very important to diagnose the symptoms before implementing any treatment or a control measure. Xylella fastidiosa in California can be transmitted by about 14 different species of insect vectors. These are the 4 main ones: the blue-green sharpshooter, Graphocephala tripunctata, glasswing sharpshooter, which is an invasive species that I'll focus today on my talk, the red-headed sharpshooter, and the green sharpshooter. So these sharpshooters, California is a long strip. So up north, on the northern side of the state, and also coastal areas, the main vector is the blue-green sharpshooter. It likes more of a cold climate. It cannot stand the heat here in the valley and also in the desert areas down south. In the Central Valley, we have the green sharpshooter and the red-headed sharpshooter. Those are mainly grass feeders, but they can do well on the other plants as well. And then in Southern California is the glasswing sharpshooter, and then also here in the Central Valley. So there's an overlap on the populations of the vectors here. Grapevines in California, they go dormant during the year. So the leaves drop off, the plant doesn't do much, physiologically speaking. So the vectors have to move out to these overwintering sites. So blue-green sharpshooters up north, they usually go to these riparian areas. So if there's a creek or a river and there's vegetation close to the river, that's where they go to overwinter. The green sharpshooters and the red-headed sharpshooters, they'll go into alfalfa fields, irrigated pastures. And the glasswing sharpshooter, they tend to prefer citrus orchards. They do quite well in citrus. They can multiply and overwinter in citrus and then move back to the grapes. The glasswing sharpshooter is a polyphagous insect, so it can feed on hundreds of plant species. So they do quite well in backyards, ornamental plants, and so on. Pierce disease in California. So we do have 2 modes of spread. So one is primary spread. We have the vectors moving into the vineyard, usually early in the spring. And if they're carrying the bacteria, they're going to drop it into the orchard. If there's enough time for a systemic infection of the plant, that plant can become a source for secondary spread. So then the vectors, they're actually established in a vineyard on the following year. They might be able to spread the bacterium. This is a classic photo from Northern California. So that's blue-green sharpshooter territory. They go into these riparian habitats, and then in the spring they start moving into the vineyard. So all these empty spots that you see in the vineyard, those are all grapevines that were removed due to Pierce disease. This is in the Central Valley. So this is a green sharpshooter and red-headed sharpshooter territory. This is a photo taken by Sandy Purcell many years ago, and where it says previously alfalfa. So alfalfa is a good host for the sharpshooters and also Xylella fastidiosa. So sort of the same principle here. Sharpshooters are moving into the vineyard, dropping the bacteria, and then the next year, the next ones will pick it up and then spread sideways. With the invasion of glasswing sharpshooter, there was the first epidemic of Pierce disease in the Southern California and Temecula caused by glasswing sharpshooter. This is an area, there's a road. I don't know if you can see a road in the center there. There are a number of wineries there. So there's a mix of vineyards, citrus orchards, and a lot of landscape plants, as you can see from the number of houses there. So glasswing sharpshooter is an invasive one. I have them here in the lab. In the lab, they can lay up to 1,000 eggs, and they can live up to 1 year here in the lab. Out in the field, we don't know, possibly a little bit shorter than that. They can feed on hundreds of plant species. So they're a polyphagous. They were first found in Southern California in Irvine in the late 1980s. And then slowly they're moving up to the north part of the state. This slide is old, it's from 2008, but I still keep it there. So the yellow areas are the glasswing sharpshooter infested areas, and the red ones, it's when they were eradicated. Excuse me. So most of those were eradicated, but just last year, there is a population well established there up north. This is in Temecula Valley, so again, they do quite well in citrus. And there was a vineyard on the bottom part of the slide, and that is gone due to Pierce disease. There were some estimates after the invasion of glass-winged sharpshooter in California that one citrus tree could host about 5,000 sharpshooters. So 5,000 sharpshooters per citrus tree. And they did that by putting parachutes over the plant and then fumigating them, counting the number of bugs. So on average, 5,000 per plant. So you can imagine the damage that it can do to a vineyard that's sitting next to citrus. This is in Southern California, about an hour and a half south of my lab here. Once you get the sharpshooters well established on the grapes, it doesn't take long, usually about a year or two, to start seeing some brown spots. I don't know if you can see some brown spots in the vineyard. There, if we take a closer look, then that's Pierce disease, and that usually leads to removal of the vineyards because it's not economically viable to maintain. The plants are not producing anymore. That particular vineyard that we're looking at, that's my colleague on the blue jacket there, Dr. Mark Sisterson. He was out there a few weeks ago, and apparently they have a cannabis hemp, I believe, planted in that vineyard. So it's not viable anymore to plant grapes in that area. So control guidelines for Pierce disease in California. These are recommendations to growers. So how do we control Pierce disease? So one of the options is exclusion of the pathogen, which is kind of hard for Pierce disease because once the grapevines get infected, there's nothing we can do about it. There's no antibiotics or anything that we can do, at least something that's economically viable to exclude the pathogen from the grapevine. So not much on the exclusion part. Avoidance of the pathogen. So if someone knows that there's a high population of vectors next door, they may avoid planting grapes in that area, which is complicated because if you do plant your vineyard, the next year someone is going to remove the almond crop and then they're going to plant a citrus crop or something that can host large numbers of insects. So, but it's possible, avoiding the pathogen. Cultural practices, not much there as well. Pruning does not work. I think in citrus, they had some success many years ago. I don't know if that's still going. I'm waiting to see Helvecio's presentation later today. But a pruning of grapevines and almond trees does not work to remove the pathogen. So that leaves controlling insect vectors and also developing disease resistance. So developing grapevines with disease resistance. Then there's also some novel methods, things that have been researched today. I don't know if I'm going to have time to go through those. But for grapevine resistance to Pierce disease, there are two ways to develop these grapevines. One is through conventional plant breeding. That's when we're making crosses of plants. And then the other one is biotechnology. So that's gene editing, transferring genes from a resistant source into the susceptible ones. At the moment, as of last week, I was talking to someone that knows a lot about the industry, and those approaches are not acceptable by the industry right now. But they are doing some research on that, and I will touch on that briefly today. So for the conventional backcross breeding, this is what we do here in my research unit. You start with the resistant source. So usually it's a Vitis arizonica that's resistant to Xylella fastidiosa, and there's some other sources too. So these are wild grapevines. They're not good for developing table grapes or for making wine. But once you start crossing them with the good ones, then eventually you can get a plant that is resistant to Xylella fastidiosa and can produce fruit that's commercially available. So you cross that first time with the vinifera, then you end up with the 50% vinifera on the first cross, then you keep crossing. With marker-assisted selection, so we do know the locus that confers resistance to Xylella fastidiosa. It's called PDR1, Pierce disease resistant locus 1. We don't know where the gene is exactly in there, so there's just a section of the genome there. So we can actually go through some of the plants and make the process much faster. So the grapevines are grown in from seeds in small vials, and you get a progeny of these crosses. Then we can collect the leaves and use those for marker-assisted selection. And then you end up with a new accession that is resistant to Xylella fastidiosa. We're actually stacking resistance to powdery mildew. Powdery mildew is a fungal pathogen in grapevines. It's actually more important than Pierce disease because of the amount of fungicide that has to be applied in a vineyard. So we can stack resistant genes into these new accessions there. So these are some accessions that were developed by a previous breeder that we had here. He retired a few years ago. But those are some good selections there. So that's a Vitis arizonica backcross 2, so only the second backcross. Seedless table grape selections with high fruit quality and resistance, shown by molecular markers there. We're also working developing raisins. So these are varieties that do well for raisin productions, and we can introgress the resistant genes into those grapevines as well. So those are accessions for raisins here. That's another one that Dr. Remy was developing. For wine grapes, most of the work has been done, well, all of the work is done at the University of California in Davis. He used to be led by Dr. Andrew Walker. He retired last year. They do have a new breeder there. And just last year, they released 5 new varieties that can be used for wine. There's one, it's kind of it's similar to a Cabernet. You cannot call it Cabernet because once you cross a Cabernet with the resistant vines, you lose the denomination there. So those are the other 4 that they were released, 2 white varieties and then 2 red ones. Again, they're all resistant to Xylella fastidiosa. Biotechnology. So this is has been done for quite a few years now. So instead of transforming the scion or modifying the genome of the scion, folks, what they're doing, they're developing rootstocks. They have been modified. One approach is to kill Xylella fastidiosa. So the rootstocks will produce these antimicrobial proteins, which will then float to the top of the scion and then kill Xylella. Another method, another approach is to suppress the symptoms. So you still have Xylella on the plants. The titer can be a little bit lower. I mean, the population density of the bacteria can be a little bit lower compared to a susceptible variety, but it reduces the symptoms on the plants. The third one is restricting movement of Xylella fastidiosa. So the rootstocks will be sending these enzymes up to through the plant that would prevent Xylella from moving from one vessel to another. So they'll be confined to one location of the vine. And the fourth approach is to restrict movement of bacteria by confusing them. They do need to communicate to form biofilm and so on, so we can confuse those. And that has also been placed into the rootstock. So they're stacking different methods to control Xylella on the vines. Again, that's not something that the industry would accept at the moment for a commercial operation for a planting, but they like to have what they call the gene shelf, just in case in the future. For the vectors, so we have, after the invasion of glass-wing sharpshooter, which pretty much changed the whole scenario here in California in the late 1980s. So if you have vineyards without glass-wing sharpshooter, one of the things that you can do is avoid planting near vector habitats. So there will be pastures, alfalfa fields, and the ditch banks as the hosts for the vectors. You can monitor the populations with sweep nets, especially for the grass feeders, and then the yellow sticky traps for the ones that are attracted to the sticky traps. Not all of them are attracted to the traps. And if you see large populations, then yes, you have to treat the vineyard with insecticides. Eliminate breeding sites for vectors so that they can probably go outside. If it's not a commercial operation next door, they can probably go in and remove some of the plants that the vectors can use. There's some limitations though. If the vineyard is located next to a pasture or alfalfa field, we cannot treat those with insecticides, or at least it's highly regulated by the state what kind of chemicals can be applied to those crops because it's going to be used as feed for cattle. Now, vineyards with glass-wing sharpshooters, so avoid planting, again, monitoring the populations. and controlling the populations in citrus orchards. They do quite well in citrus. Again, they do not cause problems in citrus, like Helvecio is going to talk about later today. Glass wing sharpshooters, they can transmit the strains that cause citrus variegated but we do not have the Xylella strains that cause CVC here in California. So it's not a problem for citrus growers. It's a problem for the vineyard next door. And then in the vineyard, one of the recommendations is to identify and remove vines with Pierce disease symptoms. So if you find a grapevine, you have to remove that as quick as possible because that one grapevine may serve as a source of inoculum for secondary spread. So it's important to monitor the vineyards as well. And the treatment for glasswing sharpshooter is usually a soil-applied neonicotinoid, imidacloprid, which apparently they cannot be used here anymore in California. Apparently they can use it if they have it in stock, but you cannot buy anymore. So I don't know what's going to happen with the sharpshooter. Well, I know what's going to happen with the sharpshooter population in the next year. What's happening now, and Pierce disease as well. It'll take about 2 years for the phone start to ring. The CDFA, California Department of Food and Agriculture. So after the invasion of glasswing sharpshooter, this program was implemented. It's coordinated by the CDFA, but in collaboration with the county ag commissioners. So these are the local district and then the universities and also USDA and the industry as well. And the goal is to slow or stop the spread of glasswing sharpshooter until we can find solutions for the disease, for example, grapevine resistance and so on. There are 5 elements to this control program. One is to contain the spread, survey and detection, a rapid response in case of new detections, outreach to alert the public, and also research. So one component is trapping. These are yellow sticky traps that you can see on the upper right side of the slide. So if you look at the maps on the bottom there, on the one on the right side, all those white circles is a location for a sticky trap. And those sticky traps are placed throughout the state. And the one on the left side, the map on the left side, you have those colorful squares and stars. We cannot read the legend here, but on this slide we can. So that we have been working on in the last few years. And once you start seeing yellow squares and blue squares, that's when things are going bad. That's about 200, 300 sharpshooters per yellow sticky trap per 2 weeks. So the sticky traps will look like the one on the bottom left side. It gets into a point that the sharpshooters don't get trapped anymore because they're landing on top of each other and then flying away. So when you see something like that, things are about to go bad. So the area-wide insecticide treatment program, that's also part of the CDFA program. So these are just some data from 2006. It hasn't changed much, so I need to update the slide. But in 2006, about 5,500 properties, so these are homes and backyards that were treated with imidacloprid and some other chemicals, and also the citrus orchards. So 15,000 hectares of citrus were treated to control glasswing sharpshooter, about 13% total. The problem is these are insecticides, they can harm natural enemies of citrus. So there are a number of pests in citrus and integrated pest management programs that can be disrupted by these insecticide applications. For urban areas, houses, this is actually my backyard. So they came last year, 2021, and they asked if they can. It's a voluntary thing. I can deny if I want, but I said go for it. It's an important issue. So on the left side, that's the notice, and my property was treated. But if you look where the arrows are, those are black arrows, a property adjacent to you was not treated to. So my neighbors declined. Everyone in my neighborhood declined to have their plants treated. So it gets hard because then you're doing like spot treatments for the sharpshooter, which doesn't work very well. You want to have area-wide applications there. There are other challenges as well. Again, the insect is a polyphagous insect, a number of plant species they can feed on. Xylella can also infect a number of plants, crop diversity, soil types, a number of things that complicate this insecticide treatment program. There's also a biological control program for glasswing sharpshooter. So that involves importation, mass production, and release of egg parasitoids. That's coordinated by the Dr. David Morgan with the CDFA in collaboration with a number of institutions there. These are some of the egg parasitoids that were released in California today. So these are females will lay their eggs inside the egg of the sharpshooter. Larvae would then eat the egg. Then you have an adult coming out of there. This is these are the two species I've been tracking. Those they're desert adapted species, and they're doing quite well actually here in the valley. So these are some data from 2009. I also need to update that slides, but we have about 7 species that were released. Number of places in the state. They have these active sites where they go back and monitor the populations, and it's more than 2 million individuals that have been released so far. And then the last part of the program is a research and extension. So that's just alerting the public not to move plant material across county lines. Keep an eye on for symptoms and sharpshooters, and notify the state government or the local government about any findings of the sharpshooter. So that's also part of the program as well. And then just to conclude, management of glasswing sharpshooter populations in California has been achieved through a partnership of federal, state, and local agriculture departments, industry, and also research institutions there. And those collaborations are essential to keep glasswing sharpshooter from spreading to other parts of the state. It's complicated because they do move in plant material, nursery material. People move from one part of the state to another. They take plants with them, so it's kind of a complicated thing. And then in the meantime, we have these research projects, which I didn't talk about today, but they're building a foundation of new information and advances to help manage glasswing sharpshooters and associated Pierce disease outbreaks. So that is my last slide. I will answer questions at the end of the last presentation there. So I'm gonna give it back to you, Dr. Ferris. I think I made it on time.
Yes, you made it on time. Thanks very much, Rodrigo, for your very nice presentation, excellent information, and updated information on glassy wing sharpshooters. So yes, we will have all the questions at the end of the 3 talks today. In the meantime, I would like to tell you that there will be in the chat an evaluation form that you could fill up after the workshop. So now I'm going to present the second speaker, who is Maria Saponari. She will be talking about disease control, and she will be talking about the Italian case, where, as everybody knows, it has been very, very important spread of Xylella in olive orchards. Maria, do you hear me?
Yes.
Okay, so great. Yeah, she will be talking about the Italian case of Xylella fastidiosa, which, as everybody knows, it has been a very important issue in the last decade. So I would like to say that Dr. Saponari started her research in plant pathology by focusing on the detection and characterization of olive and citrus infected viruses. During the past 8 years, however, she extended her scientific interest to the development of several research programs on Xylella fastidiosa, both at the national and the European level. She contributed to the first report of Xylella fastidiosa in Europe, and she also defined the genetic relatedness of the Apulian strain of Xylella fastidiosa pauca, and to identify the first ascertained vector of Xylella fastidiosa in Italy, which is Philenus spumarius. She also contributed to fulfill the Koch's postulates of Xylella fastidiosa and the olive quick decline disease, and she obtained the first complete genome of the European Union strain of Xylella fastidiosa. She coordinated the research of Xylella fastidiosa in the European Union Ponte project and led the European Union consortium of the Xylella fastidiosa project, which is the XF factors. which is the Xylella fastidiosa active containment through a multidisciplinary oriented research strategy, which just finished last year in 2021. And currently she's part of the other European research initiative targeting this pathogen, namely the H2020 MCSA RISE project, CUR-XF, and the H2020 BBI project BioVexo. So thanks very much, Maria, for talking in this workshop, and I just leave you with your presentation. Thanks very much.
Thank you. Can you hear me? Good morning, everyone, and thank you, Alberto, for the introduction and the organizer for inviting me to give this presentation. I will start by giving just an overview of the situation of Xylella in Europe. As you may know, for Europe, Xylella fastidiosa is a quite recent issue, and all the history of Xylella in Europe actually started in 2013 when the bacterium was detected in my region. Is in the southern part of Italy. I hope you can see my arrow in this circle. And the discovery of Xylella fastidiosa occurred because we were dealing with a severe new disease in olives in this region. The disease was decimating olives, mainly the old trees. And the situation appeared in 2013 very alarming because the spread was very high in the olive groves affected by the disease. It was not limited to few olive trees, but once a symptomatic tree appeared in an olive grove, soon after the symptomatic trees expanded to by interesting the whole orchard. So after different attempts to find which was the cause, we finally detected Xylella fastidiosa. So this raised lots of concern about in the European Union, because we knew very well from the literature the damage that Xylella fastidiosa can do to the crops, as Rodrigo showed for the Pierce diseases. And so soon after the reporting of this outbreak in Italy, the European Union enforced the mandatory surveillance all over the Europe. And then these are the results. Surprisingly, several other outbreaks were identified upon doing a monitoring program by inspection, sampling, and testing samples all over the Union. As you can see, I circled the main, all the outbreaks so far known in Europe. You can see that this map represents also a risk map based on the climatic suitability. So in red are the area where the climatic conditions are the most favorable for the establishment of and the persistence of the bacterium, so for an epidemics, while in the northern part of the Europe the conditions are less favorable. As you can see, most of the outbreaks and the occurrence of the bacterium have been detected in the most risky location, with the exception of an outbreak in Germany, but this was related to a single oleander potted plants. Here I reported all the year when all these new outbreaks have been identified. What is important is also to say that based on the genomic and genetic investigation that have been done on the strains identified in these different outbreaks, we discovered that actually Xylella in Europe did not arrive in 2013 or few years before, because when we discovered in 2013 the outbreak, this was the result of a 5-10 years early introduction of the bacterium. So as I indicated here, in France, and especially in Corsica and in the Balearic Island, actually the bacterium, based on the genetic information that we have developed, that the colleagues have developed based on the new tool we have now available, dated back the introduction to several decades before the discovery in Italy. However, all these outbreaks are related to different strains of Xylella fastidiosa. None of these outbreaks is related to the Apulian outbreak. So the strain that causes disease in Italy on olives is only limited to our region. In all the other outbreak, there are different strains, and generally these are less aggressive, and this is the reason why the presence and their occurrence in this area was not reported until the inspector went to specifically monitor and test for Xylella fastidiosa. So we're not the infection did not cause significant damages, so did not cause major epidemics, except the case in Italy, which still remain the worst situation of Xylella in Europe. The investigation, as I mentioned before, in all these outbreaks showed that we are dealing in Europe with different independent introductions. In fact, in this outbreak, we have strains of at least 3 subspecies of Xylella fastidiosa, so a wide genetic diversity. And when characterizing some of these strains, we found some novel genotype or sequence types, which is the general taxonomic classification used for Xylella fastidiosa, indicating that most likely in the American country, which the origin of Xylella fastidiosa, there is still some undiscovered genetic variability that then emerged when this strain were introduced in Europe. And the other aspect is that the introduction in Europe of Xylella, of course, determined the new host species. I mean, there are some plant species that were not reported as susceptible to Xylella fastidiosa in the American countries, but we found some different species infected in Europe, and this is because some are typical of the Mediterranean agroecosystem. Despite this numerous outbreak, it should be said that in any case, the European area interested by the infection and by the outbreaks or epidemics is still limited compared to the whole territory of Europe. And this is for this reason that Xylella for Europe is still categorized as a quarantine and a priority pest. And as such, there are some mandatory legislation, mandatory measures that are applied to contain and to protect the rest of the European countries and the European territories from this pest. So the first action when an outbreak is identified at European level is the so-called demarcation of the area. So the phytosanitary authorities delimitate an infected zone, which is the area that include all the infected plants that have been found. And around this infected zone, to protect the rest of the free area, the pathogen-free area, a buffer zone is delimited. And this buffer zone, as the term say, buffer, is intended to protect the rest of the territory, so to avoid that from the infected zone, the bacterium can spread further. Actually, in Europe, we have a situation in which the eradication measure applies, and situation in which there are containment. The difference is that eradication measure applies only when the outbreak is very limited and the number of species infected in the outbreak are very limited. So when we have the situation of a very early detection. So when the bacterium has not spread in the territory. Whereas and so in this case, Europe strongly support the eradication measure in the sense to remove the bacterium from the territories and to give to these territories again the status of Xylella-free area. And some example, there are some example of this eradication, like the one in Germany, but because it was related to a single potted plant, but also in Madrid there was an outbreak related to one or few olive trees that was eradicated, as well as in a nursery area in Apulia, where the bacterium was identified in potted plants and eradicated. And, while for containment measure apply when in those areas where the spread has already an endemic or epidemic pattern. And as you can see here in Corsica, for example, the whole island has several outbreaks. So Corsica is under containment, as well as our region in Italy, the Apulia region. And the Balearic Island. In Italy, here in the Apulia region, you see both measures because, as I will explain better later, we have an area where only containment applies, whereas to protect the rest of the country, there is a small boundary area where eradication applies because the aim is to protect the rest of the region and the rest of the country. Whereas in all other areas that you see here in France, Portugal, and Spain, there are still in place the eradication measure. Briefly, in what consists the eradication measure. These are the most controversial measure because are highly impacting the growers and not only the growers, because as you may know, Xylella fastidiosa affects 100 of host plants. So the measure do not involve only one plant species, one crop, but are highly impacting. So for the eradication measure, what is mandatory is to remove, when an outbreak is identified, is detected, is to remove all the infected and symptomatic plants, of course, but also all the plants in array of 50 meter. This has been updated recently. In the past, it was 100 meter. You have to remove all the plants belonging to the same plant species, regardless they were infected or not. So you can understand that in our region, this was a very impacting measure because for the olive trees, because we have olive is the main crops in where the outbreak area, and is not only a tree used as for crops, but is also a landscape trees. So it was very impacting to remove in 100 meter all the olive trees surrounding an infected plants. Now this array has been reduced to 50 meters based on the scientific knowledge that have been acquired during the past years. And the infected this infected zone is then protected by a buffer zone, the one in green, where an intensive surveillance is in place in order to intercept any new jump of the bacterium, as well as are mandatory the control of the weeds and the vector, as I will explain later. I want to mention that the demarcation of the area when an outbreak is identified in Europe, it's not has not only an impact on the crops, on the farmers, and so on, but there is also a huge impact on the nursery industry, because in the demarcated areas, there are very strict requirements for the movement of plants. So it is quite impossible for nurseries to move plants outside of these areas. So it is quite difficult to continue their business unless they switch to production under controlled condition like greenhouses or insect-proof sites. Regarding the containment measures, these include lighter measure compared to the eradication, and the aim of this measure is to live with the bacterium while continuing to be productive even in the presence of the bacterium. Actually, in the containment measure in Europe applied in our region, in Puglia, in Corsica, and as I mentioned before, in the Balearic Island. In this case, it is not mandatory to remove the infected plants, except in Apulia, where in this area, which is red, marked in red, there are no obligation for removing infected plants, except in this northern part of the infected area, where the removal of the infected plants is mandatory, and the scope, the reason is to avoid further spread toward the north of the bacterium. Whereas in the containment measure applied in Corsica and Balearic Island, this the removal of infected plants is not mandatory, except in few risky locations like close to nurseries or other important location, because these are island protected and isolated by the sea, so there is no need to have this boundary like we have in Apulia. Just quickly, the impact of Xylella in Italy on our ecosystem. The main outbreak is the one on olive in Apulia, but a few years ago, in 2018, another outbreak quite widespread was found in a promontory of the Tuscany region. The two outbreaks are not connected. We are dealing with a strain of the subspecies Pauca for olive in Apulia, whereas the outbreak in Tuscany is in an independent introduction and is caused by a strain of the subspecies multiplex. In Apulia, as I was mentioning before, this strain is causing a disaster on olive, but there are, as it is for other strain of Xylella, it is not able, this strain fortunately is not able to cause disease in almond, citrus, or grapes. As you can see here, almond are look quite healthy, where in the same olive groves that are destroyed. And also, it was also important to show that experimentally that, for example, we verified in controlled condition that the subspecies multiplex the strain of the subspecies maybe are less aggressive on olives than the pauca. So our main problem is to have a very aggressive strain in southern Italy. And this is, to summarize, the key factor of the epidemic in olive. Actually, is a quite a simple pathosystem, because at the end we have only one strain of Xylella, and is the sequence belonging to subspecies pauca, and is the sequence type 53. This is a genotype related to the coffee strain. And we have one predominant insect vector. However, we have climatic condition and a highly susceptible olive cultivars. The most ancient olive cultivars unfortunately resulted highly susceptible to the pathogen. So even if we do not have multiple strain or several insect vector, we are dealing with a very severe epidemic, because the combination of these factors are very detrimental. And looking on how and why we have this severe situation, after some year of investigation, we now know that unfortunately in the olive grove, the main pathway of spread is from olive to olive, whereas there are no evidence that there are alternative hosts that may serve as a primary source for the infection. So the infection in the olive groves progress from olive to olive. And just briefly, this is the life cycle of the insects correlated to the spread of Xylella fastidiosa. Spittlebug have generally in spring a juvenile stage on grasses. They are quite vulnerable in this stage. But as soon we approach summer and the ground vegetation dry out for because of the dry and hot season, and the spittlebugs become adults, and they found the canopies of the olive trees as their refuge and the feeding sites. And so this is all over this summer, we have this risk of infection and reinfection of olives. And in fact, if we monitor the infectivity, the number of positive specimens during this period, we reach very high value, much than I reported here, 50%. In some case, we have peaks of 80-90% of insect positive, which that for a vector-borne disease is quite very, very high. And then just briefly, here is the results of a 2 years monitoring of symptoms in an olive grove. You see how all the plants are represented by a red spot indicating a symptomatic plant. So the spread in an olive groves without management of the vector is quite rapid. Unfortunately, the spread of the bacterium is not only related to the active movement of the vector, but we found that several new outbreaks appeared because of the long jumps of the insect vector, mainly as hitchhiking on vehicles. And this evidence comes from the fact that several new outbreaks have been found close to gas stations and factories. so where a car usually stops, and so the insects are then can fly on host plants. The control measures, so in Europe, are based, as also Rodrigo explained for Pierce disease, on early detection, reduce the inoculum pressure, and vector control. For the early detection in Europe, there is in place a mandatory surveillance program. This is enforced not only in the infected and demarcated area, but also in the territories and the countries that have not yet the bacterium. And to this end, the European Food Safety Authority a couple of years ago developed a software to help the phytosanitary authority to design the monitoring, because you can imagine that with a pathogen that can infect hundreds of plants, it is quite difficult to select which one to monitor, which one to inspect, and where to inspect. So we have now this Ribes Plus software that the phytosanitary authority in Europe use to support their monitoring program. To reduce the inoculum pressure, as I explained, the removal of infected plants is mandatory in most of the outbreak, except those that are under containment. And in the demarcated area, it is forbidden to plant new orchard with species that are susceptible, unless it has been proved that they are resistant. And then the vector control. Vector control is achieved mainly through two approaches. One is to target the juvenile, which is the most recommended action, as it is more sustainable and environmentally and economically, and has higher efficacy than the control on the adults. And this is generally, as I said, the insect, the juveniles, lives on the ground vegetation. A mechanical intervention to destroy the weeds and to remove weeds is the most recommended action. Although there are several other approaches that can be used, like herbicides or other or use of host plants or preferred host plants of the insect. Of course, because not in all places we can do mechanical intervention. And these interventions are mandatory in our demarcated areas. Whereas for the control of the adults, although it is very challenging, because the adults the life is for more than 3-4 months, in which they can represent a risk being infective. What is mandatory in the demarcation area is at least a couple of treatments early in the season before the insect reach the olives and can become infective. And these are the pesticide the insecticides that are used and are authorized in our region. Regarding the attempts to cure infected plants, this as Alberto was saying, we have developed several European projects to try to find a practical solution, but all of these attempts are at experimental stage, and we have just taking advantage from what Rodrigo reported from US in grapes. So investigation are in the same directions, mainly now with the Green Deal of the European politics, mainly for using biocontrol agents, but we still are not we are at laboratory stage, and none of these has yet produced interesting results in the field. In that, there is also the problem in olive of the delivery of the products. So it is not easy, because like some product cannot be delivered by spray due to the leaves that have a different anatomy than grapes or other crops. And this is just an example. Maybe Helvecio will report about the use of N-acetylcysteine for the control of CVC in Brazil. This was one of the first product we tried also on olives, but in our condition, with this aggressive strain and with this crop, we did not get the promising results obtained for grapes. There is now a project ongoing focusing on biocontrol agents, both or natural extract, both for against the bacterium and the vector. And just to conclude, the last important research line towards the control of Xylella is the use of resistance, as Rodrigo explained, reported in grapes. Unfortunately, for olive, we are quite behind the level of the research in grapes, mainly because only recently the genome of olive has been released. And so we have several knowledge gap yet to fill. However, some of the preliminary investigation have indicated that we have a similar locus that Rodrigo mentioned before, PDR1, similar locus organization in olives, which open new routes of investigation in olive, because our initial hope was to find natural resistance in the numerous olive cultivars available, but unfortunately at the moment we do not have promising results other than in two olive cultivars, Lecchino and the selection FS17. So replanting these resistant cultivars will be very important. So to have available resistant cultivars will be very important for the restoring of the damaged area. As you can see here, an example, using a resistant cultivars, we have limited a lot the spread of the bacterium. And as for grapes, the future will be the olive breeding programs, but it's a long way. As Alberto was saying, we are starting now a new project for Xylella, and where we hope to continue to work again for developing practical solution. Thank you very much for the attention. Sorry if I used more time.
Thank you, Maria. You were almost on time, so excellent presentation, and I really enjoyed listening to you, to the updated information on the situation in Italy and the rest of Europe. As you know, the questions will be concentrated after the last talk, and the last talk will be about the disease control, the Brazilian case in this case, that will be presented by Helvecio Coletta-Filho. Helvecio, do you hear me?
Yes, Alberto, I'm here. You.
Great to see you. Dr. Helvecio has a BS in agronomy from the Sao Paulo State University, UNESP, in Brazil, and also a Master of Science in the Center for Nuclear Energy of Agriculture, SENA, University of Sao Paulo. He has also a PhD in Genetic and Molecular Biology from the University of Campinas, Unicam. He spent a sabbatical program in the Department of Environmental Sciences, Policy and Management at the University of California at Berkeley. He is currently the scientific research at the Instituto Agronómico de Campinas Citrus Research Center. His research at Jaboticabal. His research focuses on the genetics, biology, host resistance, and management of citrus pathogens, such as Xylella fastidiosa and Candidatus liberibacter, Xanthomonas citri, Philostigma citricarpa, and viruses. So thanks, Helvecio, for being with us, and now you can present your talk.
Okay, thanks, Roberto, for introduction. Okay, okay, let me see here. The slide, I think it's okay. Could you give me a tip if it's okay? Okay, so you're gonna go to talk today about the disease control in Brazil related to Xylella. You are focusing CVC in the first part, and the second part it's going to focus in olive quick decline. So my idea is put in the table some scientific evidence and some facts that it's supporting the management of CVC in São Paulo State. So just a brief introduction about Xylella. As told you in the previous talk, in the first time, the first week, and today, Xylella it's a multi-host bacterium, different plant species, and so. But talking about the economically important crops today, you're going to talk about the citrus variegate chlorosis that affect citrus sinensis, sweet orange, and of quick decline that it's affect all olive plants. Okay, just a brief idea about the different subspecies that you can find in Xylella. You have the subspecies pauca that is important for the some Brazilian crops, and also you have the multiplex present here in Brazil that is causing problem in plum, and you don't have subspecies fastidiosa that affect the grapevine and the almond has Krugner talking today. So the multiplex was the first the Xylella that was reported in Brazil, the subspecies pauca. Subspecies multiplex that it's causing serious problem in plum. So, but now our talk today will be about the Xylella in sweet orange. So also we can find Xylella in Brazil, Xylella in coffee, but the problem, the economic problem of Xylella in coffee is not so a great deal, but it's a great problem in olive decline also. For start to talk about Xylella in citrus, you try putting here some timeline of the CVC in Brazil. This is the first reported until today, and showing the progress of the studies that occurred. So the first reported was in 1987, and there was some different hypotheses. And after the disease was showed that it was graft transmissible, that was talking about a biological agent that was associated. And later, you start the works about the etiology of the problem. In 1993 was the postulate was finished, and it showed that you have a pathogenic agent that was Xylella associated with the responsible by the CVC. Okay, after the next step was provide some information about if there is some vector or not transmitting this bacteria, naturally transmitting this bacterium, and it was proved. And also some workers studying the survey in the epidemiology of the disease. So how far the disease was spread from the first from the first spot. Okay, try to understand the dynamic of the disease in the field. And so, based on these previous informations, in 1996, started to discuss about the one certified program for citrus progressive material. And it started in 1996, and after 2003, this program that I will show you briefly was mandatory. So you are talking about here in 1996 or closer to this year. Now our nursery production system was in the open, and now after 2003, it was closed. It's a protective program in the greenhouse. But in that time, you must have some material to introduce in the greenhouse. So it was a emergent selection of free mother plant was starting in the field. You selected some good plants and doing several diagnosis analysis using branch sprout by sprout, and that was select the bodywoods. And after, if the plant was negative, you protect the plant to take these shoots. And after this bodywood was used to establish in the greenhouse the mother plants, and that was the source of the buds for the nursery plants. So and so in Science 2003, this program it's mandatory in São Paulo State. All nursery plants production and different vegetative materials must be under a greenhouse proof system that was frequently checked by the absence of the for the no presence of the bacterium Xylella and others citrus pathogen. So in this time, and you're talking about here now almost in 2000, at least in 2003, the problem of the CBC in São Paulo was a huge problem. So in this time, there was the São Paulo State Agency, FAPESP, have one program to sequencing some organism to try to start the genomics in São Paulo State. And in that time, the Xylella fastidiosa was selected as a model organism to have the genome to have this genome sequenced. But it was quite important because bring to citrus or bring to citrus research some expertise in genomics, molecular biology, and bioinformatics, doing new heads for the citrus research. So it's quite important program that occur in this time. So just talking about the Xylella, the distribution of Xylella in that time, you are here. It was first reported in 1987, and quickly the disease spread for over São Paulo State. Here, just for an idea about the farmers that it was spread in São Paulo from south to north and east to west. So the disease spread quickly, and also from the São Paulo State through another some southwest region in Brazil that is located in São Paulo State to another regions, north, center, northwest, and south Brazil. So in this here you have the disease incidence of the plants. In 1996, that 10 years, almost 10 years later of the first reported, you have approximately 25% of the plants with some kind of CBC symptoms, and so the disease increased by the time, okay, year by year. So in 2000, almost 2010, 2009 was the most I can say incidence or the higher incidence of the disease in São Paulo state, almost 43% of the plants with CBC symptoms. So you're trying to put here some information about some research from so after 2012, the history will be quite different in here putting some information to try to explain what occurred with the disease after 2002. So just some biological information about the disease. So economic symptoms and economic damage different from the others disease caused by Xylella. You don't have scorched leaf in the CBC. You have small yellow spot with a brownish lesion into this small into this yellow spot, and with the disease is severe severity, there is an coalescence of the spots over here. You can see here on the coalescence and the like brown like on a scorched lesion, but it's not. It's in a high severe symptoms, and also the most damage of the Xylella in citrus. It's occur in the fruits, a great reduction of the fruit size, and with the disease so severe, these fruits, a keystone, the small one, the result juice present into the fruit. About the reductions or the damage of the disease in the fruit, if the symptom is middle symptoms, there is a significant reduction in the size, in the number of the fruits in the plant, but with strong symptoms, like you can see here, almost 70% of the 75% of the weight of the fruit was reduced, and also the number of the fruit, and also you have some damage in the juice quality. About the host plant, when you're talking about the CBC, citrus variegate chlorosis, you're just talking about this problem in sweet orange, at least for the commercial sweets, citrus varieties. Okay, so the great problem is in sweet orange. You have some reported disease, report symptoms in mandarins, some mandarin species, but the most cultivated mandarins in Brazil, for example, Citrus reticulata, it's sponca, it's quite resistant to the bacterium. In tangors, for example, I can forget it. Okay, some tangors like Ortanic, Temple, and Umatilla are susceptible, but the most cultivated Murcot tangor, that is the most cultivated tangor, that's a hybrid between sweet orange and mandarins. They are resistant also, and all the lemons, as the lime or pomelos, are also resistant to Xylella fastidiosa. And so you have some alternative hosts that you are using for only for the research, like Nicotiana tabacum, Catharanthus, and Abdopsis. They are hosts also by Xylella, but only for research interests. It's not so important for the disease epidemiology. Just for how the disease was distributed in São Paulo state from 1996 to 2009, the most incidence, the severity and the incidence of the diseases was highest in north of São Paulo state, and the few in the south part of São Paulo state. So there is a gradient of the disease from the north to south of São Paulo state. Just for looking a little bit more in this about these different regions, north and south of São Paulo state. What is the difference between this disease? The north of São Paulo state was characterized by one irregular rainfall distributions. Okay, the total of the rainfall is the same in the south, almost the same from south to north, from south and north. But in the north, there is an irregular distribution. Just for showing you this picture that we are talking this month, in this month, August, plant from the north and plant from the south part of São Paulo. The same rootstock, the same sign variety. Just see the difference, the stress caused by the water stress in these plants. Also, the temperature, the median of the temperature, it's higher in north compared to the south of São Paulo state, and this have a direct influence in the number of the generation of by year from the sharpshooters. It's important keep in mind those different environment conditions from north and south of São Paulo state. And also, it is important when you're talking about the latent infections. The latent infection is higher in the south part of São Paulo state because because we don't have good conditions for the disease expression in south of São Paulo state, but you have a good conditions for the disease expression in north part of São Paulo. So the infect plants, of course, is higher in north part of São Paulo state in that time. So, but it's not so higher in north. It is sorry, but is the infect plant is. The infect plant is the number of infect plants is low in São Paulo in south of São Paulo state, but the latent infection is higher in south of São Paulo state, and so there is a great evidence that the CBC incidence and severity was influenced by the environment. That's why we are talking about two different environments, North and São Paulo state. So epidemiology, about the epidemiology of the disease, spring and summer, that was that you have warm temperatures and the wet conditions. And so the sprout of the plant is more frequently. The success of transmission is higher in these both conditions, humid and warm seasons. Okay, about the vectors, you have different species, almost 13 different species of the vector that is able to transmit the bacteria, and also the efficiency of the transmission is quite different. And the most, at least from this information here, the most the higher transmission efficiency, it's almost 17% of the efficiency. So based on these informations, you just show here this when you call here the tetrahedron of the disease, vector disease. You only have the bacterium, the pathogenicity of the bacterium, or the virulence of the bacterium. The host, you see that all sweet orange variety are host susceptible. You have the vectors and also the environmental conditions that you have favorable to disease expressions. Based on fact, they have warm temperatures and rainfall distribution, irregular rainfall distributions. So the strategy to management the disease, remember just that you have no bactericide molecules so far. You have some bacteriostatic, but no bactericide molecules so far to deal against the bacteria into the xylem vessel. So the system, the management of the disease is based on minimizing the vector population, plant health young citrus plants, and also inoculant reductions. So that we are based on the management of the disease, the CBC in São Paulo state. You call the short to medium term of effects of these strategies. In fact, you put in some slides, you put it, but take off about the long and you call long-term effect that was related with the breeding programs that he was conducting in Citrus Center. It is the same strategy that Rodrigo Krugner told you in grapevine. Here you have some breeding programs using crossing between Murcott that was resistant and sweet orange, the pera sweet orange that is susceptible. You also biotechnology. You also have some biotechnology program that was showed by Alessandra in the first talk in the first week of this workshop, and also you have some you call mass selections that was looking in the field some variety that was tolerant to Xylella fastidiosa or tolerant to CBC. But now, today, you're talking about the short to medium term effect based on health nursery plants, inoculant reductions, minimizing the vectors population. Just remember that the early 2003, we established a system, the production of citrus plant under a vector-proof greenhouse. And just here you can see the disease incidence by the years and by the according with the by the years and according with the plant's age. You can see that after 2000, the incidence of the disease and the youngest plant was reduced significantly reduced. Okay. And also in the youngest plants, like 3 to 5 years old. So, just for emphasizing this number, here you show the numbers of the plants that you have in the orchards in São Paulo State. You can see here that from 2002, almost 2002 to today, almost 82% of the citrus plants, sweet orange plants in São Paulo State was planted after 2002. I mean that the plants, all of these plants came from the certified citrus nursery program. Okay, almost 82% of this plant citrus that you're talking about, that you are having in our orchard nowadays, came from plants produced under greenhouse, and you're talking about almost 200 million of the plants. Okay. A few parts came from the orchard that was planted in 8 years. Okay, and almost 10% of the orchard was replanted plants. Okay. Just for keeping in mind that basically all the plants that you have here in São Paulo State in the field nowadays came from the certified nursery program. Talking about the reduction in inoculant reductions, the paper that was published a long time ago, but just to remember, you can see that the most better situation to do pruning in sweet orange plant with CBC, it's when just a few leaves with symptoms was present in the branch and cut the branch almost I can't remember exactly the number, but it's almost 20 centimeters below the symptomatic plant, symptomatic leaves. Okay, and also in plants that was higher than 4 years old. Plants with symptoms with less than 2 years old, the bacteria was spread systemically for all the plants. Okay, so we recommend the uprooting of these plants is not pruning the disease branch, and also for plants with high severity of the disease, independent of the age of the plant. Also, you indicate the uprooting of the plant because the bacteria was systemically distributed in the plant. Okay. Talking about now about the minimizing the reductions, inocular reductions, and minimizing the vector population. The work that was recently published by the colleague Bassanese. Doing in the field some blocks of the plants using uprooting, different frequency, and also foliar spray against the vectors, and also put some in. Of course, this one block that was no sharp controls. Summarize the results. You can see that the uprooting every later uprooting. Okay, the disease incidence was higher compared with the uprooting every 20 years, 20 to 28 years, or 65 years. So the time of the uproot, the later time of the uproot, was directly related with the high incidence of the disease. Also, foliar spray every 20 day or every 14 days. There is no difference between these different time, but it was important to reduce the disease incidence compared with with no vectors control. So. So these facts can great evidence that just to remember that we were talking about the disease in 2010, 2011. It's almost 40% of the incidence now, and the incidence drop from 2015 until today for almost 0.5% of the plants with CVC incidence in São Paulo state. So it's for my personal point of view, the short to medium term effect, health nursery plants, inocular reductions, minimizing vector population has a great influence on those this dramatic or strong decrease of the disease CVC in São Paulo state. And I can see that this kind of the recommended activities works for CVC management, okay, because it's a fact that the disease is not more problem for the in citrus orchards, in sweet orange orchards in São Paulo state nowadays. The disease comes from almost 30% of the incidence to 0.5% in 2021. Okay, and so in this technological package, we can say this one has a great responsibility by this strong decrease of the disease. But just to remember that you have a low transmission efficiency of short shoot of Xylella fastidiosa by the sharpshooters in citrus. And also, we are thinking about the Krugner and Maria talking, the biology and the agricultural in São Paulo State, when you're talking about citrus and Xylella, it's not so complicated that you can find in California and in the southern Italy, because you don't have a lot of hosts, alternative hosts by the sharpshooters, right? And also the farmers is not small farmers, are a bit more bigger farmers. Okay. Another point that you must keep in mind that there was one increasing of the spray against psyllid insect that is transmitted by Candidatus liberibacters, the causal agent of HLB, after 2004, and directly can consequently control the vectors in Xylella vectors. And also in 2009, 2010, there was great eradications of the old orchards as consequence of the low price in that time, sweet orange low price in that time, and the growers switched to sugarcane production. So the older plants that frequently was more affected by the Xylella was removed. And also just emphasizing that the almost 82% of the orchards nowadays was planted from nursery plants produced under screenhouse. So that was, I would like to show you some evidence about what's going on with CVC in São Paulo State, the great reduction of the disease. Nowadays, when you ask for the growers, and what about the CVC, even in north or south part of São Paulo, the growers says that they have no more problem with CVC now in the orchards nowadays. The great problem is HLB, but CVC was basically was not more present in sweet orange orchards in São Paulo nowadays. So now moving the host to what's going on with olive plants in São Paulo, in Brazil nowadays. Just for you have an idea, the olive plants in southern São Paulo is planted in the mountain, surrounded by the Atlantic forest. Like you can see some pictures over here. It's a great view. So you can see here the olive plants and the mountains. So you can see here in the different pictures, different places, how you can see olive plants and mountains, olive plant and mountains. And also you can see the olive quickly decline symptoms in those plants. What you have doing with to just understand what's going on with the disease in olive plants, you show that the disease is almost spread for all of the orchards. We select some different kinds, several orchards, and in those regions, and can see the high incidence of the disease in those plants, in the Xylella also, and the almost 75% of the infect plants was colonized by just one ST, the ST16. Also, one John Lopes student shows that there is different vector species that was responsible or associated with has potential vectors of Xylella, and with high or if the transmission, if variable transmission efficiency also has you show for the citrus, okay, and you see also that when you using, for example, more variable molecular markers like SSR markers, You can see also that there is only 2 different clonal clones that was present in the infected plants. You are analyzing a great number of isolates, but you don't have the more genetically variable strains that was present in those plants. So the first question that I saw, I did was how the efficiency of the vertical transmission of Xylella by rooting. Okay, and I see that you select one plant in the field. Okay, the plant was infected by the Xylella, but was not symptomatic, and doing some different and we did routing of these different shoots, and see that the almost 57% of the nursery plant that was produced from the shoots taken from this infected plant was infected by the bacterium. But you have a long latence period. You must wait 32 months to see some symptoms in greenhouse. So you have a huge latence period in this plant. The plant was infected, but does not show the symptoms. And also, you did some questions about the pruning. The pruning could work or not by the management of disease in all of the plants. You select some plants just for a few symptoms. In fact, I did personally this one. I just select the plants that have a few leaves with symptoms, and also you collect different small branches through this main branch. Okay, and do the real-time qPCR branch that works from 30 centimeters to 3 meters below from the symptomatic branch that was located over here, and also below those symptomatic branches, and also in the root system. You take you do a broad sample in these plants, and summarizing all the results that you have that the bacteria was spread systemically over the plants. A total of 3 plants. In the 3 plants, you can see these pictures, the systematically distribution of the bacteria. So the pruning in those in the olive plants doesn't work, aiming to reduce the bacterial population in the plants. So, and also have the Xylella present in the roots. Take the home message. There is no a key recipe to silver to solve bacterial vectors problem disease, even in citrus, even in olive plant, and even of the host you can say that you can thinking about. For the CVC management strategies based in exclusion of the pathogens by using nursery plants, Xylella, pathogen-free nursery plants, inocular reduction and host plant protection works for the CVC. Based on systemic distribution of Xylella into the plants, the pruning strategy to management the olive quick decline is not recommended. And so, specifically talking about the olive quick declining, there is urgent, at least here for us, a regulatory program to production of nursery plants free of Xylella fastidiosa. So thanks for your attention, and sorry.
Thank you very much, Helvecio, for your presentation, your nice presentation. So now I think we are finished with the presentations. We are a little bit late over time, so we can start the part on questions. There has been several questions, so we can start with the first speaker. Let me check about the questions. Just a second. Okay. I will start with the questions for Rodrigo. So we can start with the first speaker. The first question was regarding how far north in the Central Valley of California has glassy wings sharpshooter invaded? In your opinion, what management strategies have been the most important for controlling the spread of Pierce disease in areas where glassy wing sharpshooter is now prevalent? These questions were made by Joao Lopez.
The glassy wing sharpshooter, there's now a new population established, probably just south of Napa Valley. For those of you not familiar with the state of California, they're trying to control that population there. It looks like it's out of control. There were some studies done that show glassy wing sharpshooter can survive and reproduce pretty much throughout the state. And I wouldn't be surprised if they could survive in Oregon and Washington, which is just north of us here. In terms of areas where we have glassy wing sharpshooter and Pierce disease, the strategies that have been working are insecticide applications to reduce the vector populations in the vineyard and also in citrus orchards next to it, and also removal of grapevines, so trying to reduce the source of inoculum there. One problem is that the sharpshooters are becoming resistant to the imidacloprid. That's the primary insecticide used to suppress their population. They have been using this chemical for more than 20 years now, year after year. And according to the scientists who did the research down in Riverside, glasswing sharpshooter can swim in a solution at full label rate of imidacloprid, and they will not die. So resistant populations are developing.
Okay, thank you very much. So the second question is about also by Marina Esteves from Centro Citricultura Silvio Moreira. She's asking among the breeding of biotechnology strategies for developing tolerant or resistant grape varieties to Pierce disease, which ones are the most promising?
So there are a number of approaches that have been developed, and those approaches have been tested individually. So these are grapevines that were transformed to produce antimicrobial proteins or to produce these confusing signals and kill the bacteria and so on. The goal is to stack them all together. So they all work well individually. The idea is to stack them into one grapevine because Xylella may overcome these resistance. Over time, depending on the pressure, they may overcome. So having different mechanisms of resistance in one plant can help. And among the strategies, conventional breeding is the only one that we have available so far. I mentioned there are 5 varieties of wine grapes that have been released. So these are available to the public. They're available to the nurseries to commercialize and use. The table grapes that we develop here, they're not available to the public yet. And the idea is to stack resistance to powdery mildew, which is an important disease for table grapes. So the idea is to stack resistance before we release these varieties to the public.
Okay, and another one for Rodrigo comes from Helvecio. He's asking how wide the control of sharpshooters is dealing with biological control using parasites, or how far parasites are being used for controlling sharpshooters in California.
So the biological control program, it's targeting organic citrus orchards and backyards in the cities. So there's no overlap with the area-wide insecticide application. So the insecticides are applied only in conventional citrus orchards, not in the organic ones. So that's when the biological controls come in, and again in the cities as well. Imidacloprid is systemic. It shouldn't be a problem to the parasitoids if it's applied to the soil. If it's applied foliar, then yes, it may have an impact on the parasitoid population.
Okay, thank you, Rodrigo. I had also a few questions for Rodrigo. You said that sticky traps were not efficient for sampling glassy wing sharpshooter, but then you showed some slides where you showed that they are being extensively used for assessing the spread of glassy wing sharpshooters. Can you tell us a little bit more about the efficiency of sticky traps?
Yeah, sorry I didn't explain that very well. The sticky traps, what I mean to say is that it doesn't work well for these grass feeders, the green sharpshooter and the red-headed sharpshooter. And I think it has to do with the behavior of the insect. So they tend to hop close to the ground, hop to the grass area. They try not to move up to the canopy unless they have to. So for those insects, the best way to sample is sweep net, just sweep in the grass. For glasswing sharpshooters and the blue-green sharpshooters, yes, they're adapted to the higher canopy and they are attracted to the sticky trap. So the sticky trap works for those species, but it does not work for the grass, low ground species. Sorry about them.
Yeah, no, it's the same story with the spittle.
Yeah.
In Europe, we found they are not very efficient to sample spittlebugs also.
Yeah, one thing about one more thing about the yellow sticky traps that I mentioned, those they have like large numbers of sharpshooters on them. The sticky traps do not monitor population density. It's more of a yes or no, the sharpshooters are here. Of course, up to a certain point, we can say, oh, there's a lot of sharpshooters here, and there are not very many there. But the sharpshooters do not tell anything about population density. It's just a yes or no.
And for spittlebugs, do you have experience with yellow sticky traps, with Philenus?
I do not have any data. I've done a lot of sticky trapping in the past. I haven't seen any spittlebugs on those sticky traps. They're usually placed up high, hanging high on the trees. And the spittlebugs, my understanding is that they tend to stay low as well. So they probably behave the same as the green and the red-headed sharpshooters.
Good, thanks. And also the last question for Rodrigo I have here is, do farmers are enforced to treat their citrus trees even if glassy wing sharpshooter is not a problem for them?
No, it is a voluntary program. So the state and the local authorities would approach the citrus growers and say, there's a high population of glasswing sharpshooters. Are you willing to spray to suppress the populations? If they say yes, then the growers would do the applications in the citrus, and then they get reimbursed from the federal government by the costs of the application. And my understanding is that those funds are not there anymore. So I don't know what's going to happen. And they're developing resistance to the chemical. So things are about to get complicated here again.
Okay, thanks. Now we will I have some questions for Maria. Does the ST53 still keep restricted to olive plants, or has it moved to any other host plants?
No, it is up to now we have in natural infection we have 35 plant species that have been found susceptible to this sequence type, to this strain. But the predominant host plant under natural condition is olive, followed by oleander and almond. These are the three main species found infected, while some ornamentals are very rarely found infected, like rosemary or lavandula, but all are also can be a host of this strain.
Okay, but actually crops, major crops, the only one is olives.
Yes, olive and almond, which is more resistant. Infection in almond are very rare, and even when the almond is infected, we do not observe severe leaf scorching or severe symptoms. The same is on cherry.
I have another question regarding the ST53. Is the ST53 related in somehow to the Brazilian strain that is causing some problems also in olives?
I think yes, in the sense that is also a coffee strain like the ST16, if I'm not wrong, but it's not very close. But both, I think, are more a coffee strain rather than a CVC strain.
But it doesn't kill the tree in a similar way as it happened with the olive quickly decline disease. No, I think it's milder symptoms. I'm not sure about that.
Maybe Helvecio can confirm, but I think there are also severe symptoms with ST16.
Okay. And another one for Maria, I also wrote. Well, let's move to some other questions made by other people. For example, about the host-resistant cultivars made by Hamza. How widely have you investigated olive trees for resistance across all the Mediterranean basin? That's what she asked.
Yeah, in the framework of the project, one of the European projects, we had a collaboration with one of the biggest olive cultivar collections, which is located in Cordoba. So we are testing more than 100 different olive cultivars from different origin in the Mediterranean. And unfortunately, not based on the symptoms, because it will take 5-10 years in this young plantation to see the real impact of the infection. But based on the rate of infection and the population size in the plant, unfortunately, we did not find any cultivar behaving like Lecino. I mean, low prevalence of the infection and low bacterial size in the plant. And I take the opportunity to answer also to the last question about the titer, the mechanism. We still don't know yet the mechanism, but what we observed is similar to what occurs in grapes. So in the resistant plant, we have low population, and the vector transmission, of course, is related to the population in the plants is very low when you use resistant plants. Is not effective, efficient.
Okay. And I think now we have a question also for Helvecio, because I think we are running out of time. Question for Helvecio by Fernando Rocha. Regarding the Xylella subspecies in citrus hosts, is there a difference in the severity of the disease in Brazil?
Well, I suppose yes. Okay, but I'm not sure about this one. It's the biology with Xylella in citrus not easy. Sometimes you have inoculations, it's success, sometimes not. Sometimes in greenhouse you have symptoms, sometimes you don't have symptoms of CVC in the greenhouse. So, but just for I don't have how to measure, but just for the symptoms and some plants that you have infected in greenhouse with different ST for a long time, you can see different kind of symptoms. Okay, but different severity of symptoms, yes. Okay, but I don't have this how to measure this information. Okay, just for one observation. Okay. Just Alberto, just for complete about the different STs in olive plants in Brazil. Yes, the ST16 is the most prevalent, and you have a huge severity of symptoms in olive plants, including the plant death.
In the olives.
The olives, yes, sorry, the olives. Sure.
Okay. Yes, because I mean the Pauca strain that you have in olives is different from the one you have in citrus, I expect, and the one that does not hold the citrus.
No. You found some citrus strains in olive plants also.
Also.
Yes, and the same sequence type and the same kind of symptoms.
So the same sequence type can affect both crops.
Yes, I suppose that all of it's for the Pauca strains. It's I can say what's universal host for the Pauca strain, because you found the citrus strain, coffee strains, all of them in olive plants.
Mm-hmm.
But the opposite not.
Okay. The one in olive, I mean, the ST53, we know now that it does not infect citrus.
Yeah.
Okay. The other question I have for Helvecio, regarding the evolution of CBC in Brazil in the last 10 years, do you think it has gone down because of the application of insecticides to contain the HLB vector, the Diaphorina city?
I think it's part of. It's not total. My point of view, the most important action was the nursery, using the nursery plants free from Xylella.
Okay.
Okay, I think if I put some numbers in those answers, okay, it's again, it's not a scientific measure scientifically, but the health nursery plants free from Xylella, it's great responsive by this one.
Okay, great. Well, I think we are 10 minutes over time, and many people, especially in Brazil, will like to have lunch. And in the US, probably they are going to have, I don't know, breakfast perhaps. Anyway, thanks very much to everybody for attending this very interesting workshop. And hope I can see some of you next week in September 5 in Piracicaba. Thanks very much, everybody, and have a nice day.
Thank you.
Bye-bye.